Welcome to this brief lecture on enzymes. Now, real quick, some characteristics on enzymes. Enzymes are generally going to be proteins. When we're looking for their name, you're going to see an enzyme generally end in ase. Once in a while you'll see zyme, but for our purposes mainly you're going to see ase. Example, lactase. Enzyme, and we'll talk more about what that name means here in a second. Third thing, enzymes are catalysts. What does that mean? That means they're able to perform a reaction without changing themselves. Okay, so they catalyze something, but they don't change themselves. They can keep doing the same thing. They're like a factory. They produce something without changing themselves. Right? A factory produces cars, but never changes itself in the process. Now, enzymes are also specific to a certain substrate. So, they are specific to substrate. What does that mean? For example, if we look back up here, we have an enzyme called lactase. Lactase is an enzyme that works only on the sugar lactose, and that should be an A. So, the enzyme has ASE, refers to enzyme. LACT refers to the sugar lactose. So it's an enzyme that breaks down lactose sugar. If you're lactose intolerant, you lack or have insufficient supply of the enzyme lactase. Now, next we're going to talk more about how enzymes work. So here's a little diagram of an enzyme process. Now, how do enzymes catalyze reactions? Why are they important? Well, what they do is they decrease activation energy. They decrease activation energy. What does that mean? Basically, it means instead of pushing a truck up a huge hill like this, so instead of trying to push that truck up that huge hill, right, which would take a lot of energy, what enzymes do is they allow you to just kind of drive it over a little hump like that. All right, so without, with. Which one's going to be easier? Definitely this one down here. So what enzymes do is they make the reaction occur easier. Quicker and easier, requires less energy, and so it's the activation energy they decrease. Now, what's the activation energy when we're looking at a chemistry aspect? When we're looking at the molecular level, the activation energy is the ability to begin the reaction by breaking bonds. Okay, so how much energy does it take to begin that reaction? How much energy does it take to break that bond and make the reaction go? That is the activation energy. So enzymes decrease that. So instead of having to drive over that hill to get everything going, it's just a little bump and you're there. Okay. So that's how enzymes make things happen quicker. We need enzymes in our body. Without enzymes, we wouldn't be able to live. We wouldn't be able to get reactions happening at the level we need them to happen or the rate we need them to happen in our body. I'm going to clear the ink from this so I can demonstrate a little bit more. Now, if we look over here on the left side at this example, down here we have the substrate. The substrate, and really it should say substrates. Okay, we have ADP, which is the molecule right here. That's adenosine diphosphate, and then we have a inorganic phosphate right there. And the enzyme is going to attach to these molecules at its active site. The active site is the site where the substrate bonds and the reaction occurs. That is the active site. Active site. Alright, the site where the substrate bonds and the reaction occurs. Now in this case, the reaction is we are creating ATP. Alright, these substrates here, enzyme is specific to this substrate, and it creates ATP. Now in that process, that enzyme doesn't change, and it can go and do it again and again and again and again. Okay, we can just keep going. The enzyme can keep going and going and going and going. Yeah, that's why we call it a catalyst. It catalyzes things, but it never changes itself. Okay, it can be reused. Now I'm going to clear the ink again here. Now, how do we control these enzymes? We go through what we call enzyme regulation, right? And some things like pesticides do this. They actually inhibit enzymes in certain insects to stop them from being able to live and reproduce and ruin the crops. And we also have enzyme regulation within our body, right? Because we don't want enzymes just performing reactions out of control. We want to control them. We want them working when we want them to work, right? So how we do that is a few different ways. Two main ways is through competitive inhibition. I'll just abbreviate inhibition there. And non-competitive inhibition, or allosteric regulation. They're the same thing. You'll just see it multiple ways in different textbooks. Another way to regulate the enzyme activity is to decrease substrate or increase substrate. Okay, so the amount of substrate is also going to affect enzyme activity. If you increase the substrate the enzyme is specific to working on, there's going to be more activity of that enzyme. If you decrease that substrate, there's going to be less activity, right? If you eat a bunch of lactose, the lactase is going to have to work harder. If you don't eat very much, lactase isn't going to work very hard. So that's another way you can regulate enzyme. So let's look more specifically at these two types. First off, let's look at the competitive inhibition. So let's say we have enzyme A here. Let's call that enzyme A. And enzyme A is specific to substrate A. Now normally substrate A is going to go and bind into the active site there. But not in this case. We got lovely old substrate B here. Substrate B is going to go bind and block the active site. It's going to block the active site and not allow A to bind. It's going to block A from binding. That decreases the enzyme activity, right? Because we're decreasing the ability of the substrate to bind to the enzyme at the active site, which is where the reaction takes place. So that's one way, besides the substrate amount that we talked about. Let's talk about one other way, which is the non-competitive or allosteric regulation. And instead of binding at the active site, so we're still dealing with enzyme A here. If I could write an A. What happens is, and let's say this is enzyme C, substrate C, sorry. A substrate's going to bind to the enzyme at a different site, and when it does that, it changes the shape of the active site. Now no longer can that one bind there because it won't fit. So when this binds, it changes the molecular shape of the active site and causes it to not be able to bind to its normal substrate. So it's where we're able to control the enzyme activity. So in this case, we would also be decreasing the activity. Alright, so the three ways we can control it: competitive, non-competitive, or allosteric, and the amount of substrate. So that wraps up our short lecture on enzymes. Make sure you get in your textbook and get in your worksheets, and good luck on the exam.